Okay, good. Start where we left off. So as you remember, this is the kind of organization. So we have an organism, and we have made of cell. In the cell of a eukaryotic cell, there's a nucleus, and that has basically all the genetic information, except for the mitochondria have their own, but besides that. And then in the nucleus are chromosomes, and on the chromosomes are loci, gene loci, where the genes are. So here we are with the genes. The chromosomes are made of a double helix, DNA, and the genetic information is coded in 4 base pairs. So we'll go on to the next and take a look at it. Okay, so what we have here is we have these backbones. Along here, right? I go up here. Okay, and those are made of sugars and phosphates. They don't carry any information. It's the stuff in between there and let's say there. Those are nitrogenous bases. Those are the things that actually code for the proteins that do all the work. Okay? So DNA really doesn't do anything. It just has kind of instructions, but there's more to it than that. There's a lot of gene regulation that's going on. So it turns out the coding region of DNA is about 2% of the total number of base pairs in our genome. And you're thinking, well, why, if it's so important? Well, a lot of it has to do with gene regulation. So as I talked to you earlier about, genes don't code for traits. They code for proteins. And the proteins are the things that form the traits, whether it's anatomy, physiology, behavior, development, any of that stuff. And so the genes themselves are turned on and turned off, but not like an on and off switch. And we'll get to that in a second. But the thing you have to take away is there are these 4 bases. AT, and then CG. These 2 bases, these pairs of bases always go together. That's the biochemistry of it. So A and T, that's adenine and thymine, and CGs, cytosine and guanine. Those are the 4 base pairs. Okay. So here we are again. As you see, we've got an A and a T goes here. These are the phosphates and the sugars, right? And then here we have C and G. An A and a G will not go together, and a C and a T will not go together. Now, we have about 3,234 megabases. That means we've basically got 3,234,000,000 bases long for our DNA, for our haploid DNA. Now, as you noticed, with DNA, there's always a complement. So on one strand, like right here, you always have a G, and the other strand is a complement of it. So if you were to split these apart, let's say for replication, it'd be easy to recreate a second strand because a C is always going to go with a G and so forth. Back in 1953, the structure of DNA was discovered. You can look at that video. It's a nice little video explaining what was going on. These are Crick and Watson. They won a Nobel Prize for it, as you can imagine. At that time, people had a feeling, perhaps, that DNA might hold the genes, that was the information encoded in the genes, but they weren't sure. And then they didn't quite know the structure, and it took some other work by some X-ray crystallographers where you can actually look at different atoms in a molecule to actually get the idea that it was a double helix. And there were a number of different people working on it at this time. These guys, their advisor was not interested. So they were doing all this on their own. While, he wanted them to do something else. But he was a fool. Anyway, so they figured it out. And then later on, Crick, who was English, Watson, American, Crick figured out the way to read the genetic code. And I talked to you about those bases. Well, if you get 3 bases long, which we'll see, you will code for an amino acid. And anybody remember what proteins are made of? Amino acids, right. So a codon is 3 base pairs, like A, C, G. That'll code for a specific amino acid. And then for a gene, you have a long string of these base pairs, and you'll get, let's say, thousands of amino acids long for each gene. Okay? Okay, so in terms of DNA replication, when you see ASE, what does it mean? What kind of protein is it?
Enzyme?
Yeah, exactly. Okay, so what happens is these 2 strands from a double helix, they start to separate, and then now we've got these single base pairs, right? Let's go here first so we can go up. So we've got these single base pairs. It could be, that might be a T, and this, of course, is a complement A, right? And then what happens is a second strand of these nucleotides, and these bases are called nucleotides. So the complement is then added on to the original parent strand, and if you actually look, here's the parent strand, and here's the new strand being formed, and the DNA polymerase that we talked about, the enzyme, is actually adding on the new bases that are complement to the original parental strand. And eventually, you end up with 2 new full strands of DNA if you're undergoing cell replication. Because each cell needs, remember, each cell has a complete genome in it, not just the genes that it needs at that period of time. Okay. Let's see. Well, I would like to look at this. We'll look at this later. But anyway, you can look at this. It takes you through the whole thing. Now, we're going to get back to this in terms of DNA and how it's actually read and transformed into proteins. But in the meantime, we're first going to talk about how it's actually used. Let's say, in this case, you've probably heard of what are called DNA fingerprinting. That means you want to find out, let's say for paternity testing, or you have a disaster or a missing person, and you want to find out who that person is. You can do what's called DNA fingerprinting. And it's a pretty easy thing to do now. In fact, there's actually a newer way to do it that's even better, but we'll go over the old way. You can get DNA everywhere. Right now, you're leaving DNA all over the place. So if a police officer came in here and decided, I'm going to put this person in jail, they'd come in here, they collect some DNA that you left, and then they plant it somewhere. No, they won't do that, hopefully. Okay, but there's tons of DNA that we leave all over the place. Now, we do something, let's say we get a small amount of DNA, a couple of cells, let's say. Well, that's a very small amount of DNA, but what we need is we need a large amount of DNA. And so we have something called polymerase chain reaction, which allows you to make copies, lots and lots and lots of copies of that DNA that you've got from the cell. And I think what I'll do is we'll run this. Let's see. Oh, that's not going to work. Okay. Hold on a second. Okay. So here's a PCR cycle, as we mentioned. We're amplifying it. We're making millions and millions of copies. And there's these variable regions of the DNA. Now, most of the DNA, for most people, is exactly the same. There are only certain regions that are variable among individuals. Now, of course, individuals have different alleles, right? So that's one variation. But there's these regions that don't necessarily code for anything that are very variable. And they're called short tandem repeats. Okay. And here we are up here. Short tandem repeats, STR. And let's say you have one individual here, and this is a repeat, a TACG repeat, okay? So you can make complements of this. So let's say this one individual in this area has 1, 2, 3, 4, 5, 6, 7, 8 repeats long. And let's say you have some more repeats. Each one of these is going to be a repeat. And this person only has 3 repeats long. Well, you would say that they're probably not related. Of course, if you had you had a strand that was 8 repeats long, you could say, well, maybe they are related. Well, of course, if you only used one STR, you'd find a lot of people. But if you have like 16 different STRs spread across all these different chromosomes, the odds of having the same number of repeats for one individual, let's say at one STR he's got 8, and another he's got 2, another he's got 15, another he's got 4, the chances over 16 of all those different variations or repeats would be very small that you'd find someone else with the exact same thing. In fact, it would be hundreds of billions of times unlikely for that to happen. So that's how we can actually identify DNA. Okay. So, and then what we do is these repeats, remember, are a certain number of repeats long. So let's say you have a repeat that's 4 letters long, 4 base pairs long, and you have, let's say, 3. We'll say right here. We'll say this is 3. No, actually, up here, that'll be 4. Okay, 3. And down here is, what is it? 1, 2, 3, 4, 5, 6, 7. And then 7. Okay. Now, oh, actually, it's the other way around. Sorry. Hold on. Let's go back. Okay. Here we go. This is B1, 2, 3, 4, 5, 6, and 7. Okay. Now, this is what's called a PCR. We'll look at this in a second. But for now, you get these bands. You have all these different people up here, right? Person 1, 2, 3, 4, 5, 6, right? Now, these bands, the heaviest or the longest strands of DNA of these repeats, let's say 7, or in this case it looks like about the longest is 6. Right up here. Okay. Let's say that's got 6 bands. That's going to be heavier than a repeat that's only, let's say, 2 repeats long. Okay? So it turns out the heavier they are, the slower they move. So up here, you have the largest fragments. Okay, these are large STRs. And down here, you have small STRs. Okay. Now remember, we're talking about diploid here, so everybody gets 1 from mom and 1 from dad. If we look at right here, we have this individual, and that's from mom, let's say, and then we'll say this is from dad. This individual is actually, mom and dad have the same number of repeats there. For this person, we'll say mom is here, and then dad is down here. Okay, so what we do is with these electrophoresis, we have this plate here, and it's got this material, and what you do is you put, let's, up in these wells here, you put a little bit of DNA. Okay, so you put some DNA there. You put some other DNA there from the other person there, right? And then you actually apply an electrical charge. And it turns out STRs, they have a charge. And so here you have a negative charge. You apply an electrical current, and the DNA is going to go towards a positive charge. Okay. And depending on how long the fragments are, they will move a certain amount. Okay. So here we have a very large fragment. It only moves a certain amount because it's heavy, so it moves more slowly. Okay, so this is a large fragment. This down here is a small fragment. Doesn't weigh as much, it moves more quickly farther down. And depending on the two fragments that you have for each of these 16 sites, you're going to get one band for your mother and one band for your father. Okay, so this is called gel electrophoresis. Now remember, we've got all this DNA because we've amplified it with PCR. So normally you wouldn't have very much DNA, but you've now amplified it, made millions and millions of copies of it so we can actually use it. In fact, with PCR now, all you need is essentially one cell with the nuclear DNA, and you can amplify all of it. Okay. So again, different size fragments, short tandem repeats, whether they're 3 long or 8 long or 15 long, they move at different rates. The heavier or longer fragments are moving more slowly, and so if the PCR goes down this way, a band up here is going to be a long fragment. A one down here is going to be a short fragment. Okay. So, out of these two, which one is shorter? Huh? Good. Exactly. That's the shorter fragment. The longer fragment is up here. Actually. Okay? Longer still. Shorter. You get the idea. Okay. Now, here's the question. We've got, these are all father 1, father 2, or father 3. We don't know who the father is. It's probably father 3. We don't know this because the baby looks a lot like the milkman. Okay? So, but we can't really be sure, right? Can't really be sure. So we're going to do a DNA test. We want to find out if the milkman is the one who's been bringing more than milk. Okay. Here we go. Now, remember, here's the baby, and one of the baby's bands is going to be from the mother. So, which band is that? Right here, right? Okay. So, this is mom's, and the baby's got that. Now, the baby also has this. So, we know that, here's these fathers again. Okay. First of all, we know that that can't be. We also know that, we want to know who's not the father. Exactly. Well, it's going to have to be two. Let's see. No. It could be, it'll actually have to be one, right? Yeah. So, it's going to have to be one is not the father, because this is the baby's band from the father, and it could be number 3, or it couldn't be number 2, right? But this band does not exist for number 1. So that's definitely, this guy didn't do it. As I said, like I told you about, the milkman. Milkman is number 3, actually, and he's the one who was, the kid looks like him. Okay? So, you can actually find out paternity-wise. Now, you can also find out, let's say you have an air disaster and all you have is body parts, right? You can get DNA from families, let's say from a mother or a father or a brother or a sister, and you know that they share half the DNA, and so they're going to share different bands with each other, and then they'll start taking these samples, amplifying them, and finding out, trying to match who goes with which. Or you could find out if some criminal was at a certain place and left DNA there, right? Okay. Now, as you know, there are 22 homologous chromosomes. That means homologous. That means the same. So, you get one from mom and one from dad. They have the same genes on them, but they might have different alleles. And then there's the 2 sex chromosomes. The X's, females have 2 X's, and males have an X and a Y. And there are these STRs called AMELs on each one. Okay? So you can see how many of these different things there are. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, and 14 and 15. Okay, they're actually going up to 16 now, but there's a newer method where they just take, well, you don't have to know it, never mind. Okay. Okay, so who's the father? Now, again, we know the baby shares this with the mom. So we want to find out who the baby shares with the father. We have F, F, F. Okay. And then whatever this one is, doesn't really matter. So we know that we have a match right here with the father. Okay? So you should be able, if I give you a gel like this, you should be able to figure out, let's say, who the father is, who the father isn't, who the baby is, whose father, is the baby's, whatever. Okay? So you'll have a target sequence, and then there'll be two individuals, let's say in this case a mom and a baby, and you want to find out who the father is. Okay. Now, as I mentioned, the FBI has chosen 13. It's a little out of date. There's actually 16 now. And they keep a database. So they use a standard set of markers of sections on different chromosomes. They use those over and over again. And they obviously, for each individual, they'll have a different number of repeats there. Okay? So you can actually then find out perhaps who the person is. Now, what would you do? What would happen if you had an identical twin? You have an identical twin, right? Let's say your identical twin is a criminal. She's not, I know. But let's say she is. And they recover DNA. Now, there's actually, you're her sister. You have the identical DNA. Okay? Essentially, you can't indict someone unless you know who exactly they are, and you can't use DNA in this case because they both have exactly the same DNA. You could basically get your sister off. They wouldn't be able to, you can't, I mean, they can't put you in jail for your sister's crime. Okay? So, essentially, if you're an identical twin, whether it's a triplet or identical twin or identical triplet, whatever, you've kind of got an out in terms of the crimes you do. So if you ever want to do a crime, I want you to think about this, okay? Because they can't get you. They literally, they can't do anything about it, okay?
Well, then how do they put them in jail?
They don't. No, they don't. You can't put someone in jail unless you actually, have the evidence, and there's contradictory evidence there. So you cannot. Supposedly, our criminal justice system errs on the side of not putting wrong people in jail. That's what they supposedly do. It's not necessarily the case. With DNA, you're more likely to not make mistakes. Like, it turns out fingerprints aren't very good. In fact, they call it forensic science, forensic science. It's not a science. It's voodoo, for the most part. And I mean it. No, it really is. They used to use bite marks, like some guy, while he was killing someone, he bit their neck, and gave them a hickey, right? Well, it turns out, and then they had all these dentists that were, experts on and lining up bite marks. It turns out they never did this until recently, where they took the dentist and they gave them bite marks that they knew who it came from, and then they asked them, who do these bite marks match? And the dentists were always wrong. They had no idea. Okay, they were experts, but this is stuff that was actually used to send people to jail. There was, you know how you can take bullets, and supposedly you can determine which gun fired the bullet. It's BS. It's not true. You can't do it. You can say, okay, what kind of gun might have shot this bullet, but there is no way you can actually say, this gun shot this bullet. I know they say they have different marks. It's BS. It doesn't exist. So all this forensic stuff is, well, they've been putting a lot of people in jail for no reason. They don't have, you can't use, fingerprints are no good either, because you have a computer look through and get some matches, and then you have to have an expert actually find and try to line them up and say, okay, this is the individual. Okay? Doesn't work. Same problem with the bite marks. Now, the only thing, DNA does work, actually. DNA actually gives you something that's reliable, that has been tested many, many, many times, and the chances of giving, let's say, a false result are more 1 in 7 billion or something like that. Okay? The other stuff is just worthless, essentially. And so they've been putting people in jail with, supposedly incriminating evidence that they really shouldn't be using. Okay. Now, if you want to read more about it, go on the internet. It's pretty bad. Okay. So, here's the DNA profile. You have 13 STR markers plus the AMEL, the one for the sex chromosomes. And if with 13, it's like 1 out of a billion, but now they bumped it up to 16 or 17, I don't know. And now they do another thing entirely where they just use single nucleotide polymorphisms, where you'll have just one change in nucleotide, and you can differentiate even more closely. So you can't, the chances of out of that are 1 in a trillion. So DNA profiling is generally useful in identifying remains or paternity or something like that. Now, have you guys heard of the Innocence Project? Anybody? Yeah. Okay. The Innocence Project. Before DNA, there's a lot of people who were locked up. Some were put on death row from fingerprint analysis or whatever, right? Well, in a lot of cases, they managed to collect DNA, those fingerprints. Well, they might have collected some DNA from the original fingerprints, or if it's a rape kit, they would have collected DNA from the semen, all that kind of stuff. And once this DNA profile stuff was starting to be used and used successfully, they went back and they started looking at people who they had DNA from that they couldn't use before because they didn't have the technology. And they started finding people, of course, that were innocent. Now, the district attorneys don't like to set people free who've been convicted. Getting conviction looks good on your resume, and actually putting the wrong people in jail doesn't look so good. And so when this started coming out, the DAs would then say, well, there were then 2 people instead of 3, instead of 1 person doing it. They wanted to keep people in jail. Finally now, there is a much stronger system in terms of exonerating people with DNA. And so a lot of people have been set free after spending 20 years in jail for a rape or a murder that they didn't commit. There's been people who were set free who were on death row. There's certainly been people on death row who were executed who didn't do it as well. So it's better now in terms of getting the right people. A lot of times with rapes, you find out that, oh, the person who actually did it is in jail for another rape, right? Usually rapists are kind of serial rapists. So this DNA stuff has actually set a lot of people free, because you can obviously, if you have DNA and it doesn't match, you can eliminate that person as a suspect. Okay? So, saves suspects time and hassle, like, 25 years in prison. It saves them a lot of time. Okay? Okay, so what could the baby's STR pattern actually look like if I were to give you this? Well, remember, there's two. Each person has two. Mom has two. Dad has two. Okay? So we could actually go through and we could say, okay, this is B1, B2, B3, and B4. Okay? Now, I'll show you what it looks like, but right now we could actually figure it out. Here's Mom, and we'll take her first one right up here. Right? In fact, we'll put there. Okay? We know the baby could have this, right? And we know the baby could also have, let's say, Dad's first one, right? Right here. And so that's what baby number 1 will look like. Now, baby number 2, go back. We'll take same mom again. And now we have the different dad. And so here is what baby number 2 looks like. Then we take mom again. And we do it again. And here's mom. Right? And we'll say the upper dad. And then mom again. And the lower dad. Okay? So you can actually figure, this is a baby can have one of 4 patterns for me, each pair of parents. Because they all have 2. And you can see how it works out. So, again, mom and dad and mom and dad from this upper one, right? And then, and the same thing that we just did. Now, there are certain things that are not possible. Remember, each person has 2 bands, one from mom and one from dad. So, you can't have 3 bands, nor can you have 4 bands. Okay? Nor can you just have 1 band. Now, occasionally, you can have a situation, let's say, where here's mom, here's dad, and they both have the same band. So you can get a double band at this area, right? And we see that a lot, and I'll show you a few examples. Okay. Okay, so some things aren't possible. Let's see. Okay. Oh, well, sorry, this one. This is dad, right? The baby is identical to dad. The only way that's possible if the dad had an identical twin. Okay? They would share the exact same bands. So dad has an identical twin, right? But that could never be the dad's baby unless mom had a band at the same place that dad had a band as well. Okay. Let's see, what am I at? And that's about the end of that. Okay, now, what I'll do is, what time? Good, we got a little bit of time. I'm going to open up some top hat, so you guys should answer in class here. And then we'll continue on after about 5 or 10 minutes, something like that.